of your classroom. So, question booklet, page 5. Cellulose and glycogen. Now, differences. Jessica, you start us off with one. Yep, so that's made of, I'm not writing the full answers, but give yourself 1 mark if you wrote both of these down. So, you need to say cellulose consists of beta glucose, glycogen made of alpha glucose. Anybody talk about the bonds in it? Okay, so Cameron, what do you say?
Cellulose is linked together by 1,4 glycosidic bonds, and glycogen is linked together by 1,4 and 1,6 glycosidic bonds.
Yeah, very good. So yes, you need that detail. That's brilliant. Thanks very much. Okay, that's great. Thank you. What else have we got? Caitlin, what have you got there?
Cellulose is straight unbranched chain and then glycogen branch.
Yeah, so straight unbranched chains. And this is basically very branched. Okay. Now, sometimes they ask for structural differences. So all of those are structural differences because they're talking about the structure. Now, they didn't ask just for structural. But if you got a question that asked you about the structural differences, it would have to be something like that about the shape of the molecule. Is it branched or not? What monosaccharides does it contain? What type of bonds does it contain? But they just asked you for 4 differences. So you could have said that cellulose is used for structural support, or its function is structural support. And glycogen, it's an energy storage molecule. If you said cellulose is found in plants and glycogen was found in animals, that would also get you a mark, okay? So this is an older question, so they're a wee bit more fussy now, and they do, maybe it might be in a 3-mark question, give 3 structural differences, and they would only have taken the first 3. But as I said, that's not what the questions were this time. And then on page 6, you have to work out which is which. So you've got A, B, and C, alpha, alpha, beta. So straight away, you've got a good idea that C is Jasmine Broderson. Sorry?
Cellulose.
Yeah, cellulose, because it's the only beta-glucose polysaccharide you know. A and B, the other Jasmine, what could they be? What's made of alpha-glucose? Okay, so we've got glycogen, we've got, well, the polysaccharide, we just call it a starch, which consists of amylose and amylopectin. Which one is more branched, Jessica? Out of all of those that we talked about, well, just compare starch and glycogen, which one is more branched? Glycogen. So if we look at the shape of the polysaccharide, short, highly branched chains, that's going to be glycogen. And A is going to be starch. If you wrote amylopectin, that's also okay. It does tell you that A is found in plants, so that obviously links in with starch. Glycogen, you know it's found in animals. It's also found in fungi. And cellulose is only found in plants. Function, these two, energy store. Cellulose, structural support. Now, obviously we're going to do a little bit more theory and so on, but we'll just make a note of your homework, what you can do. Now, it looks like an awful lot, but some of those have only a few words on them. Okay. Okay, so last night you were filling in the first sort of page and a half. By pages, I'm talking about like the half pages you've got there. So proteins. Hopefully you've remembered that examples of proteins are enzymes, hemoglobin. There are proteins that will facilitate diffusion, and they're called carrier protein molecules and cell membranes. We'll look at those later when we look at the structure of the cell. Antibodies. There's a lot of talk about antibodies and antigens there. Well, antibodies are protein structures. And then obviously there are structural proteins for support, things like keratin and collagen. You've heard of those, and like beauticians will talk a lot about those kind of things, about your structure of your skin and stopping wrinkles from happening and all that. So, I mean, proteins are important for enzymes, for controlling all of the reactions that happen in your body, but also they're there for a lot of structural reasons as well. You may remember, well, you're going to tell me what proteins are made of. Subunits called, Jacob, what did you write there?
Amino acids.
Yep, so you should have amino acids there. And how many different ones are there, Lloyd?
20.
Yep, 20. Now, they're not going to ask you that. But there are just 20 different amino acids, and yet there's millions of different proteins. So, if we look at this diagram here, you learnt there's a carboxylic acid group. So hopefully you recognise the COOH, and that's the carboxylic acid. So you can circle that and mark that on your diagram. NH2 is the amino group. And then R is not an element. Like C is carbon and H is hydrogen. R is short for the residue group. Now, every amino acid has got these bits here, okay? So every amino acid has got all of this. But what makes one of the 20 amino acids different from the other 20 amino acids is what is joined on at this point here. So you might have CH2OH. That'll be one amino acid. You might have different combinations, you might have much more longer structures. But as far as we're concerned, you just write R down there, and it's the bit that differs. So, some of these R groups may contain things like phosphorus, they might have sulfur, so there are other elements there, but basically all amino acids contain carbon, hydrogen and oxygen and nitrogen, so all proteins contain those elements as well. So that's an amino acid. When two amino acids join together, what's formed? What's the structure that's formed, Karis? A dipeptide. Good. And just like before, if we look at the diagram, there's our two amino acids. We can see we've got H and O and H there, so that comes out. And we've got water, and that just joins over there to make this bond. And what's the name of the bond that's formed, Amy? Can you remember? Peptide bond. So on the diagram, you can label that as a peptide bond. Now, and then if you get a whole chain of these, so for example, insulin has got about 100, I can't remember, I think it's 153 amino acids or something like that in a big long chain. That's called a polypeptide. So up here, missing words are, we've got dipeptide, the bond is called a peptide bond. It takes place, the reaction always takes place between the OH group of one amino acid and the H group of the other. So a peptide is when you get several amino acids joining together. So a dipeptide is when you get two. A peptide is when you've got a small group. And then if you've got hundreds joined together, it's a polypeptide. And as we said, this is a condensation reaction. So what I want you to do is put an arrow going down and going up. And on the way down, we're writing condensation. And on the way up, we're writing hydrolysis. Now, probably my water would have been better coming out the other side. And in hydrolysis, obviously, we need to add water in there to allow hydrolysis to occur. So the next bit then talks about organisation of proteins. So just like we have 680 odd pupils in the school, they're organised into groups. And we've got different levels. So, you could say, okay, we've got junior school pupils, sort of middle school pupils, and sixth form pupils, okay? So like years 7, 8, 9, 10, junior school. Middle school would be 4th and 5th form, and then 6th form. So we could have little groups. And within those groups, we could look and we could separate them into year 8, year 9, year 10. So there's different sort of levels of organisation within a school. And the same is true for proteins. Now, I want to just take in a few things here. Where are we? Why is this not showing? Can't work. March. Right, tell you what. I'll sign up there. Structure of proteins. Okay, we're in now. So that molecule there is, you've all seen, like, chemistry teachers' models with, the wee balls and so on, the stick. So that's hexokinase. That is, it ends in ASE. So what kind of protein is it, or what kind of molecule is it, do you think? Enzyme. So it's got over about 6,000 atoms in it. Okay, so some proteins are really complex. And we've said they've got different levels of structure. So we talk about the primary structure. What do you think the next structure is going to be? Secondary, and the next? Tertiary. Any guesses what the next one is? Quaternary, all right. So that sort of makes sense. So primary structure is basically the sequence of amino acids in the polypeptide chain. So you need to write that down in your notes. Levels of protein organization. Primary structure is the sequence of amino acids in the polypeptide chain. Now, we've just said that when two amino acids join together, the bond that joins them together is a peptide bond. So the primary structure, you've got a little diagram there showing you sort of lots of circles. Inside the circles, there's different names of amino acids. And basically that is, the the primary structure, you need to know that they're held together by peptide bonds, all right? So I would highlight that in the text underneath the diagram. Primary structure, the sequence of amino acids in a polypeptide chain, and the bonds, peptide bonds. So on my diagram on the board there, I've got a peptide that has got 129 amino acids. Now, we learnt that some ions are positively charged and some are negatively charged. And in this chain, there will be, the different amino acids will have different R groups. And we said sometimes there's sulfur, sometimes there's, lots of complex arrangements of molecules. So some of them will be positively charged, some will be negatively charged. What kind of bond forms then? We talked about it when we were saying about water molecules being attracted to each other. What's the name for that type of bond? Hydrogen bond. So primary structure is simply just the order of the amino acids, but then because some of those will be attracted to each other, some of them will form this kind of coil shape, and that's described as an alpha helix. And some of them will take on this kind of zigzaggy shape, which is called a beta pleated sheet. Okay, so in terms of your notes, so down here, it could coil to form an alpha helix, and that's the most common secondary structure, or it could fold into a beta pleated sheet. So this is the alpha helix structure, and this is the beta pleated sheet. We've said they're held together by hydrogen bonds. Okay, so that you've got hydrogen bonds linking them. That's a permanent thing. Whenever the amino acids are joined together, when the protein's been made, they're joined in the right order, and that order is determined by the DNA. And so they're put in the right order, and then, as I said, some will be positively charged, some will be negatively charged, and that will cause it to coil or to fold. And that's permanent. So you need to know that the secondary structure is, what we'll say, secondary structure, we'll write should really have said the coiling, but coiling first. The coiling or folding of the polypeptide into, I'll put the alpha helix first and then. Tertiary structure. So we've said we've got all these amino acids joined together. And then they'll coil up. But if, for example, there's a sulfur group there and there's a sulfur group there, they will be, brilliant. Excellent.
Does that go home?
It does, yeah.
It does. Mr. Kelly might have a box for it there to keep